Yeah. Good afternoon, everyone. I'm speaking on the prospect of hemp protein meal in marine fish species. This is the picture of hemp that we use in the project, and I will talk. Okay. Okay. So we look at hemp because the product is relatively new in terms of using it for aquaculture in production of feed. So one of the reasons is this because we're looking at alternative ingredients to replace fish meal. We know fish meal is not really sustainable in terms of aqua feed industry. So we're looking at accurate nutritive information on these new ingredients. In 2019, 2020, the state of Florida passed the Farm Bill to make a farming of hemp in the state legal. And one of the things that came to mind was that if this product is legal and we have opportunity to farm fish in the state and other states across the US, what happens to the byproduct? Because the meal is a product of, it's a byproduct after the extraction of oil. So that was the reason why this project became important in terms of looking at the marine fishes. So the main objective, we have other specific objective, was to determine the survival and growth characteristics of Florida pompano and red drum when fish meal is being replaced. We look at it in two of these marine fishes, Florida pompano and red drum, as you can see them on the slide. So we look into 7 diets. The 7 diet contains 1 control, which is a fish meal-based diet. And the 6 test diet had 3 graded level of hemp meal. So 5%, 10%, and 15%. These 3 levels of hemp meal are with or without taurine, which made it up to 7 diet. So 5% with taurine or without taurine up to the 15%. So to the result, we did a lot of looking at several other parameters, but looking at the growth, this was really significant in the sense that we saw that when we increased the M from 5 to 15, we saw a growth, negative growth response from Florida pompano. However, when we added the taurine, we saw that the negative growth response was improved as a result of taurine supplementation. We can compare it to the one that I earlier showed. And when we look at this growth response, we compare it to the control, which is completely fish meal. The 5% hemp meal with taurine performed better, the best diet across the 7. So we saw that addition of replacement of hemp meal with 5% hemp and taurine performed better than fish meal. Looking at the red drum, also we saw similar result. From 5 to 15, negative, that is a reduction in growth. With the addition of this hemp, we saw that the growth was improved. Comparing this with the control, we saw that the 5% protein with taurine performed better in red drum. So we can see in both fishes, we saw 5% hemp protein with additional taurine better. So that means we could go into looking at replacing fish meal if this ingredient is available, is approved for aquaculture. We had looked at several other things in terms of the microbia, which I will not be able to present, but all the analysis about completing. So we have 2 publications in preparation within the next few months. Other output in terms of this research, we have 1 student oral presentation, which I will be elaborating more in the next slide, at the ARD symposium meeting, which is the Association of Research Director meeting. We have 2 oral presentations at the Aquaculture America Conference. We also have 1 oral presentation at the Women of the Water Conference. Elaborating more on the first presentation, which was a undergraduate student presentation. The oral presentation was presented by Zinaya Alvarez. She won the second prize oral presentation. Here she was, standing with her prize or with her award. Presently, she's entering into her third year. She's presently into third year veterinary student at the University of Minnesota. Another student on the project is Nathaniel Hector. He is presently a USDA scholar. She has won the award for summer internship at the USDA office in Minnesota. And he has been admitted to early program into the veterinary medicine program at the same University of Minnesota. We have other students that were trained on the project. Because of time, I won't be able to talk more about them, but during this discussion, I'm open to that. Two graduate students, Hallelujah defended in the summer. Jonathan is getting ready to do his defense. I would like to acknowledge the opportunity from USDA on the project. It has really been a great journey with both collaborators and many people that participated on the project, especially the Abba Branch Oceanographic Institute staff, where this work in terms of the culturing work was conducted, because here at FAMU, we have the freshwater system. So I can mention many of them that participated in terms of sampling, in terms of helping. And also the FAMU Aquaculture Lab, my students, staff, and many people that work with the work. So thank you for listening, and I wait for questions during the discussion.